first of all, thanks for the invitation to come here. It's my third or fourth time, I think, in Vancouver. Unfortunately, I can't spend the whole weekend here because it's Mother's Day on Sunday and I have to go home. So this is a very quick trip, but it's a great pleasure to be back here. I'm on a scientific advisory for a number of different Genome Canada proposals or funded projects, and so I've seen the operation pretty close hand, and I would say it's a high-quality organization. So I'm really privileged to be back here again. So what Gabe asked me to do is give you an overview of how genomics is used inside a seed company, a big agricultural seed company. And I'm going to try and do that at a pretty high level. It's not a scientific, technical presentation. It's more of an overview of how things are used. But I want to take a step back and just sort of talk about one of the issues for why I think a lot of people are in agricultural research in the first place. And I think this is certainly true for myself. This is why I'm in agriculture, is that there is a real contribution to society for growing food. And growing food is a necessary thing for our society to survive. In the last 50 years or so, it's kind of strange. I was looking at this screen, but the slides are over here. So, the last 50 years, the population of the planet has more than doubled. And in the next 35 years, we're going to add the equivalent of 3 Chinas to the current population. So obviously this comes with a need for more food getting to people, whether that's more food production or better food transportation, better infrastructure is a very valid debate. But there absolutely needs to be change in the way that we handle production of food. And it's not just the growing population that impacts this. We have the demand factor, which is obviously the numbers of people, but also the nature of the societies they're living in, the increasing wealth, the increasing demand for animal protein instead of plant protein. All of that puts more and more strain and stress on a plant productivity scenario. And on top of that demand factor, you've got challenges and constraints coming from just the planet as a whole. We have less arable land that's available to grow crops on. We have less water to grow those crops with. We have less farmers to grow those crops with. Many populations are moving to these big megacities, particularly in the developing world. People are fleeing the rural areas, going to the big cities. So who is going to be producing the food for all these people in the cities? So all of these constraints lead to a real point in time where there is a great opportunity for innovation in agriculture to really make a difference. And innovation comes from many different places. It comes from management practices, it comes from technology, and it comes from things like genomics. This is a slide showing the productivity of corn, average yields for corn over the last, since 1930. So you see how yields have gone up fivefold, more than fivefold since 1930. That has been achieved through a number of different means. So there's certainly management practices. There's better genetics of the corn seed. Plants are more able to withstand the stress of being planted closer together. So therefore we can have increased plant density on the field. You can produce more seed. You have more access to crop protection products like herbicides and insecticides that keep the farmer able to keep his crop viable through to the end of the season. So all of these they all play a part in increasing productivity. You see we're only at an average of 160 bushels per acre right now. And the projections are that really to supply the quantity of food necessary, we have to get to about 300 bushels per acre. Now that's an average. There are currently farmers producing 300 bushels per acre. So we know the genetics can do it. We know the plants can grow at that density. We know the fields can produce that much food. But that's the outliers. We need to move the entire average up there. And so technology as a whole is needed to come into this game. And one of the technologies that really enables some of the discoveries that can lead to these increases is genomics. Now, I missed a lot of the morning session, but I get the sense there's a lot of non-plant people in the room based on the topics this morning. So quick raise of hands if you believe that humans are the main focus of your work. How about plants are the main focus? Oh, wow, okay. So more people than I expected with the plants. So I wanted to give a quick background on some aspects of plant genomics that I think might be enlightening for how the groups of scientists work together. So for people in the genomics world, in the beginning means when the genomes were first published, right? So I worked on plant genomes starting from 1995, but in the beginning it was really around 2000. The human genome draft was published in 2000. Most of you probably don't know that in that same year, the finished high-quality plant genome, Arabidopsis, published in the same year. But what is interesting, I think, is to follow these two worlds after this point. So after these two publications, what happened in those two communities? So in the human side, what happened is it spawned a whole bunch of genome projects, all with one goal, and that was to feed information into understanding the human genome and understanding the human species. Okay, so a very spread out and then help bring information back. What happened in the plant side of things is a very different outcome. So we had the first genome, and what happened was those people who did the first genome went off and did multiple other genomes, but every single one of those has a different goal, and that was to serve another small community of people working on a different crop. And so what we end up with is this first side of different communities, each with their own genomes, each with their own version of gbrowse, their own version of standards, their own version of bioinformatics pipelines, and their own databases. And so you end up with a very mishmash of different resources, a much reduced critical mass to go ask for funding. And I think if you were to compare the funding on these two sides of the screen, you would recognize that there really is an opportunity for doing things much better on the plant side than what we have done in the past. We've had some great successes with some great genome projects, but they're all individual projects, and we don't really have much of a food production genomics standardization. So we also have, along with those societal challenges in terms of scientists' societal challenges, we also have some actual biological challenges working with plant genomes. We have some very large genomes to deal with. So sunflower, so Lauren Riesberg, I think, is somewhere in the room, works on sunflower, and that is a genome that's about human genome size, or maybe a little bit bigger. But there are others that are much bigger. So there's the wheat genome, shown here is the poppy genome with 30 gigabases, and then the winner of the prize is this lily, or a relative of the lily, is maybe 150 gigabases in size. So that's the haploid genome size. Add on top of that, we have polyploidy, which is pretty common across many cultivated crops. So shown here is strawberry, canola, potatoes, and banana, all polyploids. So it adds more complexity to the bioinformatic analysis if you try to understand these genomes. Most of the size for these large genomes comes from transposons. And these are very active, mobile elements that create havoc if you're trying to understand it from a sequence analysis standpoint between two individuals. So transposon work and research is probably much more active in the plant domain than it is in the mammalian domain. And it's important because it contributes to a high level of intraspecific diversity. Transposons move around a lot. And shown here is just the point that the intraspecific nucleotide diversity for maize is higher than the diversity between human and chimps. So there's a lot of diversity, often mediated by transposons. Added on top of the nucleotide diversity, you also have the massive structural diversity, such that something is on chromosome 2 in one line of maize, there's no guarantee it's going to be on chromosome 2 in another line, because things are moving around all the time. And then lastly, the evolution of plant genomes has had a lot of whole genome duplications, which adds even further to the complexity. We've got some very large gene families with very So now I'm going to shift gears and just talk about how we use genomics within the company setting. So I work for AgroSciences, and we sell two types of products. We sell bags of seed, and we sell bottles of chemicals. So the chemicals help the farmer control the pests that try to destroy his crop, and the bags of seed obviously help produce his crop. But we don't sell genomics. We don't sell genomics products. We don't sell technology in that sense. But we try and use it, so we're consumers of genome technology that you guys are producing. My group is essentially a core genomics group that helps the researchers around the company. So many of you have seen this slide, or some variation of this slide. This is the next-gen sequencing revolution in action. Shown here is the price of data generation. And trying to explain this to an organization whose model is how many bags of seed do I sell, and you're trying to tell them you need to use this technology. It's very hard to get the message across. And so I was chatting with a friend of mine, and he had this wonderful thought experiment to try and demonstrate how much data this technology can produce. And so I've taken it and modified it a little bit. And it's really asking the question, if you take the Illumina NextSeq sequencer, which is the latest model, benchtop, $250,000, can do a generation, run in about a day. So how much would it cost to print out the data that that machine generates? Mm-hmm. So for that single day run, you could actually buy 5 new sequences. So that's how much data. So these kinds of examples are necessary to really bring an organization that doesn't live this, breathe this every day, bring it into really thinking and sort of conceptualizing what a paradigm shift it's going to take inside a company to be able to utilize this kind of information. And so that's part of my job, is to really try and bring along a bunch of naysayers into seeing the benefits of this technology. So we have, my group basically handles the thing on the top here, which is data generation through to analysis. And we apply these tools that many of you are very familiar with to our core crops. So we work on corn and soybean, sunflower, canola, and cotton. That's the seed, that's the bag of seeds that we sell, right? But we also, because I told you we sell products that help farmers control the pests, we also work on, my group has to understand the genomes of lots of weeds, lots of insects, fungi, and bacteria. So we have a pretty diverse species base that we have to work with. And then thinking about what types of applications do we use genomics for in the company, I came up with basically 4 boxes. One is the baseline genomic information. So that's the Genome project, if you like, for a new species. And many of our core crops already have genomes, so that's not really an issue. But when you come across a new pest or a new insect that has never been worked on, you suddenly realize the value and how nice it is to have all those genome resources already prepared for you. Because if you have to start from scratch, it's actually not so nice. So that's one category, and it's absolutely essential. Another category, which I think probably in academic research at least, we don't think about that much, and that is using these technologies as essentially an assay to get a result about one sample. And usually that result is to make a decision on the basis of that result. It's not investigation, it's not information gathering for knowledge, it's just decision making. And I think that's one of the key differences between working in industry and academia, is that